हेलो हेलो हेलो, आलिन आवाज आ रही क्या? हेलो 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 हेलो, आवाज आ रही क्या? हेलो 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 हेलो, आवाज आ रही है ना? हेलो हेलो,
आ रही ना सर आवाज?
हाँ तो अच्छा, जवाब देने का ना, आ रही बोल के। ओके
हाँ वो माइक बंद था हमारा।
ओके, चालू करो। ठीक है, देखो, हाँ, नेक्स्ट यूनिट और नेक्स्ट वीट, द स्टडी ऑफ एयरबॉर्न पैथोजन। इन दिस वीट यू हैव टू स्टडी द मेथड्स एंड एप्लीकेशन। इसमें क्या स्टडी करने का आपको? मेथड्स एंड एप्लीकेशन। कहीं के द स्टडी ऑफ एयरबॉर्न पैथोजन। अब एयरबॉर्न, एयर, व्हाट इज मीन बाय एयरबॉर्न पैथोजन? Airborne pathogen, the pathogen which are present in the atmosphere. These are the airborne pathogen. Okay. Okay. See here in next slide. Or what is the application of this aerobiology? Okay. These are the applications. See here, to isolate the aeromycoflora. Actually, the aeromycoflora means the pathogens which are present in the atmosphere was collect air continuous air sampler and petrified exports method from the atmosphere. This is the first application. Huh? Next thing, to identify the spores of aeromycoflora. To identify the spores which are present in the atmosphere. The third is to determine the prevalence of spores in different seasons. Definitely, in different season, in atmosphere, the spores or the organisms are different in different seasons. So here, to determine the prevalence of spores in different seasons. And last application is to determine the impact of fungal spores on vegetable, fruits and human health. Okay, these are the applications of aerobiology. Okay, or through this branch of aerobiology, we can study these things. We can isolate the pathogen from the atmosphere. We can identify the spores which are present in the atmosphere, and we can determine the prevalence of spores present in different seasons. So definitely in different season, the different spores are present, or different bio components in the atmosphere are present. And last is to determine the impact of fungal spores on vegetable, fruits and human health. Okay, so these are the applications of aerobiology. Our next thing, huh? See here, the aerobiology. What is aerobiology? Actually, it is a interdisciplinary science of applied significance. The biological components in the atmospheres are considered to be the most important biopollutants, which insist the disease to crop, human health and fruit, vegetable deterioration and air pollution. Aerobiological investigation shows two Identify biological components which are allergic in nature in air, causing allergic disease in human beings. And air pollution. And air pollution. The atmospheric air contains variety of biological components transported through the air current are the main source of human allergy. And air pollution. The biological components are toxic and responsible for causing serious health hazard disease in human beings and create lot of environmental pollution in the entire surrounding of atmosphere. The fungal spores, pollen grains, fungal hyphae, algae, insect part, protozoan, cyst, plant parts, and unclassified groups were found in present in almost all seasons. The presence of biological components in the environment causes serious health hazard problem in human beings. Therefore, cleanliness should be maintained. So, actually, it is an interdisciplinary science of applied significance. In this aerobiology, we have to study what are the different biocomponents which are present in the air or which are present in the atmosphere, which are present in the atmosphere. Okay, these components includes, see here, this component includes fungal spores, pollen grains, fungal hyphae, alga, algae, insect part, protozoan cyst, plant parts, and unclassified groups. So these things are present in the atmosphere. Actually, these things they causes the different problems in case of human being, in case of plants and fruits, or in case of atmosphere. Okay, they make the pollution, they make the air pollutions. Okay, so next let's see here the biodeterioration. See here the biodeterioration. Actually, the biodeterioration means sarna of vegetable, fruits, and allergic disease in human being is serious problem encountered everywhere. The biodeterioration includes rotting, damage, and spoilage. The analysis of composition of aeromycoflora and their effects show various serious problems like vegetable and fruit biodeterioration, air pollution, number of allergic diseases in human being. First, Tilak in 1980 and 1982. Here, S.T. Tilak actually he is a professor at Dr. Babasaheb Ambedkar University, Aurangabad, and he is father of aerobiology. Actually, he is a father of actually. In 1980 and 82, S.T. Tilak developed one instrument, and the name of the instrument is Tilak Air Sampler. And the second instrument that is the roto rod air sampler, and he got the Nobel Prize in this field for the development and the study of this aerobiology first time in India. So here in India, this S.T. Tilak actually S.T. Tilak sir is from our region. Internationally, he worked on aerobiology and he got the Nobel Prize from the government. Okay, see here in the next slide, huh? How we can study the aerobiology. Here, these are the methods. These are the methods. The first method is the collection of the spores by Tilak air sampler. By Tilak air sampler. See here, you see this is the Tilak air sampler. This is the Tilak air sampler. Tilak air sampler. Actually, how it was, I will show in previous slide. This is the Tilak air sampler. Tilak air sampler. See here how it works. This volumetric Tilak air sampler with 75% collection efficiency and greater retention capacity, it runs on an electrical power supply that is 230 AC and provides continuous sampling for a week, that is 8 days. The sampler consists of two mechanical systems, the clock system and the exhaust fan system. The electrical clock fitted at the bottom in the instrument which is synchronized with the motor of exhaust fan fitted at the top. The air is sucked in the tilak air sampler through exhaust tube at the rate of 5 liters per minute. Infringing on cellophane tape on the outer surface of the circular drum. This drum is fitted on the clock system and rotates in the anti-clockwise manner, and it completes one rotation in 8 days, thus giving a trace of catches for 8 days. White petroleum jelly in thin coat was slightly coated on the cellophane tape. Adhesive in property, cellophane tape is 1.5 centimeter in width and stuck on slowly rotating drum. Thus, this coated tape faces the orifice of outward projecting tube 0.5 centimeter away from it. The drum rotates continuously, slowly when the air sample is operated. This is a in a clockwise manner, which gives continuous air sampling for 8 days. The drum has making marking of day and night, dividing it into 16 components. Thus, such each segment of cellophane tape is carefully removed from the drum and mounted on clean glass slide with a glycerin jelly as wanted. The care is taken that the microorganisms deposited on the tape are not disturbed while handling. During air sampling, the air is sucked in through the orifice tube or strikes forcibly over the adhesive cellophane tape. The exhaust fan fitted in the circular opening in the lead of atmosphere forces the air out of the collection chamber, creating a vacuum in the collecting chamber. An exhaust hole 6.2 centimeter left in the lid of the apparatus. So, see here, actually, this is the Tilak air sampler. Okay. In this Tilak air sampler, there are two systems. One is exhaust fan and second is drum. Actually, here the drum is inside this, and this drum has 16 marks. Each mark is of 12 horse. And for completing this rotation, for completing this drum, it requires 8 days. That is 8 days, day and night. That includes the 8 days. From this, actually here there is opening and it runs on current. As the drum rotates, one part of from the 16 is completed in the 12 hours. So in 12 hours, which types of spores are present in the air we can identify or we can collect. Actually, this is the photographs of weed market. In this weed market, you have collected number of spores which are present in the atmosphere. See here, this is the next method. See here, what happens in next method? See, this is the filtry fluid expose method. Filtry fluid expose method. In this filtry fluid, there is a medium. That is a potato dextrose agar. PDA medium. Potato dextrose agar. This is the fungal spores cultivated on PDA. Fluid incubated at room temperature for 5 to 7 days. Okay, this filtry fluid contains PDA medium. PDA medium. That is a potato dextrose agar. PDA medium. Okay, these PDA mediums, when after they are in petri fillets, and this medium is kept for 5 to 7 days after the exposing of this petri fillet in the atmosphere, from which we can collect the spores. So this is the second method. After 5 to 7 days, some colonies of the fungal spores, bacterial colonies, algal colonies, and different biocomponents are seen on this petri fluid. From this, we can identify the presence of spores which are present in the atmosphere. So this is the second method. So these two methods are very important and very common. One is by Tilak air sampler, or second is by Petri fillet expose method, and that Petri fillet contains the PDA medium. Okay, this is the photograph. I have told you this is the photographs of video market. And here, see here, this is the collected spores. This is the collected spore. Near about 33 spores. Near about 33 spores, which are present in different seasons. These spores include Aspergillus, Alternaria, Cladosporium, Fusarium, Mucor, Penicillium, Carolaria, Rhizophus, Lacinidia, Petrosporium, Leptosporia, Sporetima. So these are the spores. These are 33 spores which are collected from the weed city, particularly from the vegetable and fruit orchid. So in this manner, in atmosphere, there are large number of spores, fungal spores, and other biocomponents are present. But here we are interested, as we are interested for the collection of the fungal spores. So we collect the, or we identify these spores. So this is the spores. In rainy season, the flush sign indicate that these are the presence of these spores. And the Venus strain shows these are the absent. So in rainy season, these things are present. These spores are present. This is the winter season and this is the summer season. Generally, in winter season, the spore is more because the fungal growth is very high in winter season. Very high in winter season. The next slide. See here, this is a graphical representation of recorded fungal spore. Here, in rainy season, the percentage is more as compared to this two. But generally, in winter season, the spores are more. So the present study conducted that through the personal meeting. So this is the second thing. In this second thing, or second, for the study of allergic diseases, we meet personal with a few fruit seller, vegetable sellers. The 80% pupils are affected with these allergic diseases in different manners. Allergic diseases in different manners. So these spores which are present in the atmosphere causes number of allergic diseases. For example, asthma, eczema, dermatitis, mycosis, urticaria, and aspergillosis. These are very common diseases in at marketplaces. So, in marketplaces, it is necessary to maintain the cleanliness. We seen that there is no hygienic condition of marketplaces. So, due to that, different allergic diseases are seen. Think so. These are the photographs. Human diseases caused by fungal spores. So, these are the very common human diseases. This is the dermatitis. This is the nail of leg. It shows the damage, and this is the cold or allergic diseases due to pathogens which are present in the air. So these are the different diseases which are caused by the biocomponents which are present in the atmosphere. So from this, we concluded that the present studies was proved that the fungal spores in market environment are responsible for air pollution, deterioration of fruits, vegetables, and some allergic diseases in human beings like nasal bronchial rhinitis, skin, eye problem, and hair problem. So, due to the biocomponents which are present in the atmosphere, they are responsible for this disease, nasal bronchial rhinitis, skin, eye problem, and ear problems. Okay, these are the references. And thank you. So, see here, I will go first slide. So, this is the study of airborne pathogen methods and application. So, again, I will explain in short. There are two methods. One is collection of the spores via tilak air sampler. The second method is petri fluid expose method. Petri fluid expose method. And these are the applications of the aerobiology. These are the applications of aerobiology. If you have any doubt, you can ask the question. Still now, in examination, the question was not asked on this video, but sometimes it may be asked. Okay? आवाज आ रही है सबको? हेलो 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 जोया आवाज आ रही? हेलो हेलो. So this is all about the aerobiology that includes study of airborne pathogen methods and application. Thank you. Thank you very much. If you have any doubt, you can ask me or you can comment on YouTube.